Welcome back. Let's start out this part of the lecture by reviewing the organelles and their functions. Here's a list of those organelles that I pointed out in the last section. So let's review their functions just briefly. First of all, we have the nucleus. The function of the nucleus, as we said before, as we mentioned before, is information storage. It houses the DNA, our genetic information that makes us who we are. There's ribosomes. Ribosomes function in protein synthesis. Proteins are one of the types of organic molecules. And it's in our ribosomes within our cells, that's where these proteins are built. So synthesis means to build or to make something. We have rough ER. The rough ER synthesizes proteins that are either going to be exported from the cell or that are going to end up in the plasma membrane. Now, I said here synthesize protein, but it is actually the ribosomes that synthesize proteins, but these ribosomes are located on the rough ER. So the rough ER is called rough ER because there's these ribosomes, those are the little red dots in the picture from before, all over attached to this endoplasmic reticulum, so they call it the rough ER. So the ribosomes on the rough ER synthesize the proteins, but these proteins end up inside the rough ER, and they're manipulated and prepared to either be exported or end up in the plasma membrane. The ribosomes aren't all located on the rough ER. Some of the ribosomes are just floating around in the cytosol within the cell. We have smooth ER. Smooth ER and rough ER are made of the same membranous material, but their function is a little bit different. The smooth ER synthesizes lipids, so fats, and then also does some detoxification of things like alcohol and those sorts of poisonous substances. Okay, then we also have, sorry, I had to fix that. The smooth ER actually doesn't detoxify alcohol, but drugs and different things like that. Okay, the Golgi apparatus. The Golgi apparatus functions in transport and packaging of proteins that are made in the rough ER. Then we have the lysosome. The lysosome functions in digestion. It breaks down or digests larger molecules within the cell. The peroxisome, it also functions in detoxification. And this is where alcohol is detoxified. And then also free radicals that are formed in metabolism are also detoxified in the peroxisome. And then there's some mitochondria. This is often called the powerhouse of the cell. This is where ATP, that we talked about in the last lecture, the energy currency of our cells, this is where the ATP is produced inside our cells. So there are the different organelles and their functions. I want to look a little bit more about a few of these. The first one being the rough endoplasmic reticulum. So if you look at this picture here, this is the rough ER, this is the membrane here. Notice this is a ribosome. The ribosome synthesizes proteins. Here's a protein that's being built by this ribosome. So this ribosome will come and attach to the rough ER, build the protein into the rough ER. Within the rough ER, notice that there were these little green sticks here. Those are carbohydrates that were put onto the protein. And then also remember that structure of a protein determines its function. So even though the ribosome will build the protein, the ribosome doesn't determine that structure of the protein. Part of that folding and creating of a structure is done within the rough ER. So you see that there are some modifications made, protein has a certain structure, and then the rough ER is going to package that into what we call a vesicle, which is an outbudding of the membrane in the rough ER, and then that vesicle will then travel. So let's watch it move here. Okay, so now we have the protein being packaged into the vesicle, the transport vesicle. It buds off of the rough ER, and now this transport vesicle is now going to move to the Golgi apparatus. So this protein never leaves this membrane. The rough ER, just part of the rough ER buds off and forms this little package, this little transport vesicle that will then go to the Golgi apparatus. So here's our rough ER. Here's the next picture here. We have our rough ER, and now we have our Golgi apparatus. Notice here, we'll notice first the proteins attached to the rough ER. Then we have these vesicles here that are budding off the rough ER, and then they come and they attach or become part of the Golgi apparatus. So here's our transport vesicle coming down. Notice how it comes and it buds off the rough ER, attaches onto the Golgi apparatus. Then in the Golgi apparatus, these proteins, these little dots here, are going to be further modified and prepared to have one of three things happen to them. So the first thing that can happen to these proteins that leave the rough ER and come to the Golgi apparatus, remember the Golgi apparatus is all about transport and packaging. After the proteins are packaged, they're put into another transport vesicle. This transport vesicle comes to the plasma membrane, where it attaches to that membrane, and it's going to release its contents outside of the cell. This is the first thing that can happen to those proteins. Next possibility, the same main function here, the same main pathway that we just saw. We have proteins coming from the rough ER coming into the Golgi apparatus, but this time, instead of secreting the contents outside the cell, this time the contents of this vesicle here are located on the outside of the vesicle. So you see that the vesicle looks empty, but you have all these little things sticking off the ends. And this vesicle here is going to go and it's going to attach to the plasma membrane, but these components, these proteins here, are going to end up as part of the plasma membrane. So instead of releasing these outside the cell, this little vesicle here, it's made of membrane, just like the plasma membrane. So it attaches to the plasma membrane and basically becomes part of the plasma membrane. And then these proteins that are attached to it also become part of the plasma membrane. That's the second possibility here. Okay, the third pathway is that these proteins are going to be loaded into a vesicle. And in this case, these proteins are going to be enzymes, digestive enzymes. And so now this transport vesicle here has now become a lysosome. So lysosomes are organelles that function in digestion. They're full of protein enzymes that digest things, break things down. And this lysosome is created as a vesicle that buds off of the Golgi apparatus. This vesicle, or this lysosome, can then fuse with old organelles in the cell, foreign substances, or other things, foreign invaders, such as bacteria, things that may have invaded the cell, and it can fuse with those larger organelles or larger membrane-bound vesicles, and then the digestive enzymes inside the lysosome will enter that vesicle there, and it'll digest whatever's inside it. Peroxisomes, I'll mention peroxisomes a little bit more here as well. So we've now seen lysosomes. Peroxisomes function in detoxification, but they do not come from the Golgi apparatus. They reproduce by splitting themselves in half to create more peroxisomes. But they're also a vesicle-looking organelle that's floating around in the cytosol inside the cell. So now we've looked at the nucleus, we've seen the ribosomes, we've seen the rough ER, the smooth ER. I mentioned the smooth ER, we didn't really look at the smooth ER, but it synthesizes lipids, etc. We've looked at the Golgi apparatus. We've seen lysosomes. These are the major parts of the cell and their functions within that cell. Remember, though, that every cell in our body does not necessarily have all these organelles or have them in the same abundance as other cells, depending on the functions of those cells. So let's look at a few different examples here. Here is a type of cell here, and this is a neuron, and one of the characteristics of a neuron is that it has a certain shape. You have the main body of the cell body here, and then you have an axon as well as things called dendrites coming off of a neuron. This is all formed by the cytoskeleton. So the shape of the cell, such as a neuron or a digestive cell or a muscle cell, all of these cells have different shapes. These shapes are created by the cytoskeleton, which is found on the inside of the cell. In this picture here, the cytoskeleton has been illuminated so that you can see the different types of fibers and how they're giving that cell its certain structure. So some cells may have a lot of cytoskeleton, may have very unique shapes. Other cells, like red blood cells, are just going to be more circular shape as opposed to having a very specialized shape. Notice here we have some different shaped cells. We have red blood cells. Red blood cells, that I mentioned before, don't have a nucleus. They basically have no organelles. They're a specialized cell, and erythrocyte is the scientific or the biological name for a red blood cell. They have no organelles because of their specific function. They don't need any of those organelles in order to function. Notice this cell here has both rough ER and a Golgi apparatus and a nucleus, but has certain structures that are a little bit different. Here are some square or cuboidal shaped epithelial cells. They have a nucleus, they have intermediate filaments, cytoskeleton, that are giving them their shape. Depending on what type of cell this might be, what type of epithelial cell, it may have different types of organelles. So, for example, inside the pancreas, the pancreas's job is, one of its jobs is to produce a lot of enzymes, which are proteins. So enzymes are proteins. And so within the pancreas, the cells of the pancreas, you'd expect to have a lot of rough ER and Golgi apparati that are going to help in producing those proteins and transporting them to where they need to be. Here's another type of cell. We have some muscle cells here, and you can see they're different shapes. They have nuclei. We have skeletal muscle, which is long and thin. We have smooth muscle, which is more spindle-shaped. We have things called fat cells, adipose tissue. They have a nucleus, but notice the function of a fat cell here is mainly storage, and so all of its organelles are going to be pushed to the very outer edge of the cell in order to give room for this large storage of the fat droplet. Notice here the nucleus is smashed up against the side of the cell. We have macrophages, a type of white blood cell. So the white blood cell, its job is to catch bacteria, other foreign substances, digest them, remove them from the body. So as you would expect, you're going to find lysosomes inside these macrophages that allow them to digest these different attackers of our body. And then also, it needs to be mobile. It needs to be able to move around the body. And its cytoskeleton and these things called pseudopods are what allow it to do that. Here's another example of a neuron. Notice the nucleus way back here. And then we have these long processes, axons and dendrites that are formed by the cytoskeleton. And then here's another type of cell, a sperm cell, which is another very modified cell because it has no organelles except it has mitochondria that are located in this area of the sperm cell that allow this flagellum, the tail-like structure, to move. It provides that ATP that allows that movement. And then we have a nucleus, the genetic information. And that's basically all that makes up a sperm cell. So depending on the type of cell, you're going to find different organelles and different abundances. Another example, your liver. Your liver both produces a lot of cholesterol. It produces the cholesterol that we need in our body. Also does a lot of detoxification. So you expect to find a lot of smooth ER within our liver cells. So depending on the cell and its function and where it's found, it may differ from this generalized structure of a cell that we've seen so far.